I'm going to discuss about antibody labeling technique, which is a very important technique because labeled antibodies are used in diagnostic kits. And how to label these antibodies is by a different technique that involves labeling with the radioisotope or labeling with enzyme or fluorescent probe or chemiluminescent probe. In this case, one need to take care that the antibody FAB arm, that is antigen binding arm, is left free for binding of the antigen, and only the non-antigen binding arm, that is FC arm of the antibody, is labeled with the probes. Let us see what these A, B stand for and the label stands for. In this diagram, the A stands for biotin. Biotin is vitamin B. It's a soluble molecule. It has a strong affinity to the FC arm of the antibody. The FC arm of the antibody can bind to biotin molecule. And B is another molecule, that is streptavidin, or it could be avidin. The streptavidin molecule is obtained from the bacteria, Streptococcus bacteria, and it is a protein molecule which has a strong affinity for biotin. It binds to biotin. Similarly, avidin is another protein molecule, can be obtained from the hen's egg or bird's egg. The avidin word has come from birds, aves, A-V-E-S. This avidin protein also has strong affinity to biotin, just like the way the streptavidin has the affinity. Either of them can be used to bind to biotin. Now, before binding the streptavidin or avidin to biotin, these two molecules can be independently made to react with the label. Here in this case, label stands for either enzyme or a fluorescent molecule or a chemiluminescent molecule. Streptavidin or avidin is made to react with enzyme, and that enzyme is linked to streptavidin through a linker molecule, where one can use glutaraldehyde, which helps in binding of the enzyme to the streptavidin. Similarly, the fluorescent probe or fluorescent molecule can be made to react with streptavidin by the linker molecule. Similarly, chemiluminescent molecule can be made to react with the streptavidin. Once they are reacted upon and they are added to the sample where the antibody is readily bound to biotin molecule, the streptavidin goes and binds to biotin, and it gives you the linking like this. A stands for biotin, which is readily bound to the FC arm. B stands for streptavidin or avidin, which is linked to the indicator molecule. Generally, the indicator labels that are used for developing the kits are as follows. As far as radioisotope is concerned, predominantly it is I-125, which is a radioisotope, or I-131. I stands for iodine. Iodine isotope 125 or iodine 131 isotope is used. It is capable of recognizing the tyrosine residues in the antibody and readily bind to the tyrosine residues without any complication. So radioisotope I-125 and I-131 are predominantly used. For developing radioimmunoassay kits, where the antibody is now labeled with radioisotope for radioimmunoassay. As far as indicator enzyme is concerned, enzyme could be alkaline phosphatase or beta galactosidase or horseradish peroxidase. These are the three common enzymes which are predominantly used in most of the immunodiagnostic kits. And they have their own substrates and they can develop indicator reactions, which we will be seeing in the future lecture. So alkaline phosphatase or beta galactosidase or horseradish peroxidase. The source for alkaline phosphatase is predominantly from the bacteria. Similarly, the beta galactosidase is also predominantly from the bacteria, obtained from the bacteria, and horseradish peroxidase is from radish, extracted from radish. The fluorescent probe as an indicator, when used, it would either consist of fluorescein isothiocyanate, which shows fluorescence, or rhodamin, which is also another molecule which shows fluorescence. Both of these molecules show different types of fluorescence. When chemiluminescent probe is used as an indicator label, the material used here is adamantyl dioxetane phosphate. Adamantyl dioxetane phosphate. And this adamantyl dioxetane phosphate, on release of phosphate due to the reaction of alkaline phosphatase, will start luminescence. When it is adamantyl dioxetane, it will start showing luminescence. So these are the common indicator labels that are used for labeling the antibody. It is also seen that in the immunodiagnostic kits, sometimes the antibodies used in the diagnosis, which are not labeled, may be used for trapping the antigen by immobilizing these antibodies on an inert surface. The inert solid surface would act as a substrate to which the antibody has to be bound. To make the antibody bind to the inert surface, a special protein is used which is called as protein A. And this protein A is derived from the cell wall of Staphylococcus aureus bacteria. And the protein A has a strong affinity to the carboxyl terminal, COH terminal of FCR of the antibody. The CO terminal or COH terminal is present at the tip of the FCR. Therefore, the antibody that binds to the inert surface with its Fc arm, due to its affinity to the protein A, which is readily coated on the inert solid surface, would be in such a manner that the anchored antibody will have its FAB arm, that is these two arms, FAB arms, free to readily bind to the antigen that is added, for the detection. So, the protein A, which is readily coated on the inert surface, acts as a sticky molecule and attracts the Fc arm of the antibody to bind to it, so that the FAB arm of the antibody, that is antigen binding arm, is free. Such immobilized antibodies are used in radioimmunoassay, ELISA, and immunofluorescence kits. Thank you.